Okay, well, today we are on exercise 4, and specifically we're going to be covering molecular transport, and we're going to be talking about diffusion, we're going to be talking about dialysis, and then we're going to be also covering osmosis in potato plants. The plasmolysis part, please refer to your lab introduction, the video, refer to your lab manual. Unfortunately, we didn't have access to Elodia, the plant that we use, so we could not do this demonstration for you. We're going to start again in your lab introduction. Your instructor should have discussed with you the cell membrane, the properties of the cell membrane, namely mainly talking about the semi-permeability of the cell membrane and its structure. So as I said, we're just going to be doing a couple of demonstrations so you have an idea what you would have done in lab. And since we are actually in lab today, we're going to turn the camera to show you how the lab looks like. And you guys would have been working in groups of 4. So each table would seat 4 students, and this would be your workstation. But again, we're trying to do the best we can do under the current situation, but I wanted to give you a slight idea about how the labs look like. Okay, we're going to start with the diffusion demonstration. It's not in your lab manual, but we decided we're going to do it because it's pretty easy to set up. Just reminding you, when we say diffusion, what are we talking about? So diffusion is the movement of particles, of molecules, from area of high concentration to an area of lower concentration until equilibrium is reached. So today we're going to be using just plain water and a drop of food coloring, and we're going to see what's going to happen when we put a drop of food coloring in the water. So initially when you do it, you have a drop, and then you see that the dye is starting to spread, and give it a minute or 2 or a second or 2, then you're going to see that the whole water turns green. And actually, this is what diffusion is. So when you drop the dye, it was concentrated in one part of the beaker, and then the molecules of the dye started spreading until now you see it disperse throughout the whole beaker. And again, this is diffusion. Movement of molecules from area of high concentration to an area of low concentration. This is also what we call molecules moving down the concentration gradient. And as you probably learned in lecture, this is a passive form of movement, meaning no energy is required. Okay guys, we're going to be setting up our dialysis experiment, and what we use for this experiment is a dialysis tube. Before soaking it in water, it's really kind of a little bit tough, not easy to open, not easy to deal with. So in order to use this dialysis tube, we need to soak it in water. We soak it for about an hour or so. And when you soak it in water, you can see it becomes much softer and much easier to deal with. So why are we using a dialysis tube? The dialysis tube is actually simulating the cell membrane. So it behaves like a cell membrane, meaning it is semi-permeable. And again, if you recall, the term semi-permeability means certain molecules can cross the cell membrane, and in this case, certain molecules can cross the dialysis bag, but not others. The cell membrane is semi-permeable because of its structure, and in this case, the dialysis tube is semi-permeable because it has microscopic pores all over, and the size of those pores gives the semi-permeability, meaning the size of the pores is going to allow certain molecule to cross, but not others. So this is the story of the dialysis tube. What we're going to put in the dialysis tube, we have a mixture of starch and glucose. So it's a solution that contains starch and glucose. And as you guys recall from chapter 3, when you learned about biological molecules, the glucose is actually the monomer of starch. So glucose molecules are much smaller compared to the starch molecules. Before we move on and set up our dialysis tube, I will ask you a question. So if we put starch and glucose in this tube and we soak this tube in water, which molecule do you think will be leaving the dialysis tube and going into the water that's surrounding it? Let's think about it for a second. Again, we did say we have glucose and starch, and glucose is much smaller. So my hypothesis is going to be that when dialysis occurs, and when we give time for the dialysis to take place, I'm going to say the glucose molecule is more likely to leave the tube compared to the starch. How are we going to test for that? How are we going to set this whole thing? Again, we're going to take one of these dialysis tubes, as you know, the one we had soaked for a while. We will tie one end of the tube, and now we're going to put some of that starch glucose solution, and these tubes can be a little bit hard to open, so I just put it in water, just like what you do with your grocery bags. Sometimes you need little wetness to open them, so we're doing the same. And now my tube is open, so I can put my solution in it. And again, here is the solution, as I said, starch and glucose. So I'm gonna fill the tube halfway, and I'm gonna tell you, explain why we do halfway, why we don't fill it the whole way. So with the pipetter, I'm putting some of this solution in. I'm gonna put a little bit more. Okay, I think I have plenty for what I need. So I'm gonna tie the bag on the other end, and I'm gonna use a piece of twine to do so. Okay. So now I got sugar and starch all over my dialysis bag. I want to make sure I cut the extra twine. Some instructors like to use dental floss or, rubber band. I like to use a piece of twine. So this is the dialysis bag with the solution in it. Before I do anything with it, I need to really rinse it well and dry it. I want to make sure there is no sugar or starch on the outside of the bag. So all I would have to do, as I said, just make sure you rinse it well. And I dry it. And I also make sure my bag is not leaking, it's all sealed well. And I'm gonna take this dialysis bag and I'm gonna weigh it. And then we're gonna discuss why are we weighing this dialysis bag. Okay, so can you please record the number that you see on the scale? And always remember, when you're writing any sort of measurements, any sort of numbers, you need your units. So please write the number with the unit next to it. We need this number for later in order to discuss whether osmosis occurred or not. Okay, let me first, so this is the bag weighed. And now we're gonna just put it in water, and we're gonna let it sit in water for about a couple of hours. The reason we are doing that, because we're gonna see if osmosis is taking place. What is osmosis? Just a quick reminder. Osmosis is diffusion of water molecules, which means water molecules are gonna move from an area where they are more concentrated to an area of less concentration. And again, in this case, I'm talking about movement of water molecules, not solutes, just the water. So if you are looking at this beaker, we have the dialysis tube containing the starch and the glucose. And this bag is soaking in tap water. Per unit volume, where do we have more water molecules? Where is the water more concentrated? Inside the dialysis bag or in the beaker? What do you think? Well, the beaker has no solutes in it. It has no starch. It has no sugar. So in this case, what would you think of the movement of water? Is the water going to go inside the dialysis tube or is going to leave the dialysis tube? Based on what you learned about osmosis in lecture, my hypothesis is the water will move inside the dialysis tube, and if I let it sit for a couple of hours and I weigh the tube again, I am expecting an increase in the weight of the tube, which to me indicates that osmosis occurred and water moved from the beaker inside the dialysis tube. So that's the osmosis part of this experiment. Now the dialysis part of this experiment, so in the dialysis tube we have starch and we have glucose. Which molecule do you think is going to be leaving the dialysis tube and going into the beaker, into the water in the beaker? Again, when we discussed the dialysis tube, we said it had microscopic pores, and those microscopic pores are going to allow smaller molecules to cross rather than larger molecules. So in this case, my hypothesis is the glucose will, or some of the glucose molecules will leave the dialysis tube, and we're going to find some glucose in the water in the beaker. That's dialysis. Now, how are we going to test for that? How do we know that starch or glucose left the dialysis bag? We have two tests that we will run. We have one solution. As you can see in here, it's really that nice blue color. We call it Benedict's solution. And this solution is used to test for the presence of glucose or simple sugars in general. So it's used to test for glucose. And once we have our experiment going on for a couple of hours, we're going to do our testing, but there is going to be a color change. The color will change from this blue to almost orange juice color if glucose is present. And we'll talk a little bit more about this test once we get to this point. But for now, let's remember that this particular solution, Benedict, is used to test for the presence of glucose. We're going to use iodine, and I want you to see the color. It's like brownish amber color. If I add iodine to a solution that contains starch, then we're going to see that this amber color will turn into really deep blue color, like almost jeans, dark jeans color. So dark blue color indicates the presence of starch in a solution. So anyway, these are the tests that we are going to use. And after 2 hours, the bag is soaking, the dialysis bag is soaking. We'll continue with our experiment, and I'll explain more the tests that we are using. And we're waiting 2 hours. we're gonna set up the osmosis in potato experiment. And again, as we go through this experiment, we're gonna explain the steps, the objectives, and then come up with some hypotheses. And I will tell you how we are running this particular experiment. Okay, our last setup, we're gonna demonstrate. We're gonna do osmosis in potato plants, meaning we're gonna see the process of osmosis using potatoes. So we're gonna start. First, let's explain what's the objective, what are we using in this experiment. Yes, of course, we're using potatoes. We are using different concentration of sodium chloride. So we have 0.9% sodium chloride, 2.5%, and 5%. And then we have our control treatment, which is distilled water, meaning it has no salt in it at all. We have 4 beakers that we have labeled with water, 0.9%, and so on. So we have all our solutions already measured, and we put them in beakers. And we did put 20 milliliters of each one of these solutions in the water. And as you recall from lab 1, we use graduated cylinders to get an accurate volume. So I measured 20 mils of each using graduated cylinder. And of course, I don't mix graduated cylinders, so I have one for water, one for the 0.9, one for the 5, and one for the 2.5%. Okay, so I have my salt solutions ready, and we're gonna take some potato samples. And again, before I do that, let's come up with some hypotheses. So we're gonna take potato and soak it in different salt solutions. So we have 0% salt, 0.9, 2.5, and 5%. So when we think about our potato samples, we're gonna start by weighing them. So we're gonna record the weight before we put them in the solutions. We're gonna let them sit for about 45 minutes, and then we're gonna weigh those potato pieces again. So what do you think is going to happen? Well, first treatment I'm gonna start with is the 0.9%. Why do you think we went through the trouble of preparing 0.9 and not 1% salt? Do you think there is any significance to that? Well, I will tell you that 0.9% is actually considered isotonic environment to the plant cells, because in the plant cells and other cells, there are some solutes. There is some salt in there. So 0.9% is considered isotonic environment. And I'm assuming that by now you understand the difference between isotonic, hypertonic, and hypotonic environments. Okay, so if this is isotonic, 0.9%, what do you think water is compared to the plant cells? Correct. That's going to be considered a hypotonic environment. So now the cells, when you put your potatoes in water, the plant cells are placed in hypotonic environment. With the 2.5 and the 5%, you have higher solute concentration outside the cells. So obviously these are hypertonic solutions. Okay. So if we soak our potatoes and come back and weigh them again, what do you think is going to happen? These are our hypotheses. We're going to write one for each one of these treatments. So if I put the potato in water and 45 minutes later, do you think the weight of the potato piece is going to go up, down, or is it going to stay the same? When cells are placed in hypotonic environment, what do we expect? Correct. We expect the cells to gain weight, meaning water is going to go inside the cells, So that piece of potato, we expect it to increase in size and to gain weight. That's my hypothesis. We're going to see if it's true or not after 45 minutes. In the 0.9, the isotonic environment, what do we expect is going to happen to the weight of the potato core? Since we said it is isotonic, so water will be moving in the potato cells and out of the potato cells at about the same rate, so we expect the weight to remain the same. You can have a slight increase, a slight decrease in the weight, but it won't be really significant. And now for these last two solutions, the 2.5 and the 5%, which are hypertonic, we're going to expect the potato cores to lose weight, so they're going to decrease in weight. These are the hypotheses. We're going to set up the experiment. We're going to, as I said, weigh the potato cores before, let them soak 45 minutes, and then we'll weigh them 45 minutes later, and we'll see if our hypothesis is true or false for each one of these treatments. The way we're going to collect our potato pieces, we use a corer. You can see this thing, it has two parts to it. One of those cylinders has a larger diameter compared to the other. The one we use to cut through the potato, we use the large diameter corer. The smaller one we use to actually push the piece of potato out. Because if you end up using the smaller diameter one to get your piece, then the potato is stuck, and then you have to be in lab forever trying to clean it. I won't let you leave unless you get it out. And you'll find out how hard that is. Okay, so we're going to use the large diameter cylinder in this case. We never put the potato on our hand because, yes, this corer can actually hurt you. You put your potato on the flat surface, and we go straight down, and it's actually pretty easy and satisfying to do it in a way. So you get a really nice piece of potato. I bring with me always some water because I want the potatoes to be hydrated as I'm collecting all my specimens. We're going to get 4 cores because we have 4 treatments. So 2, oops, 3, and 1 more. Okay, we're going to remember then when we're setting up an experiment, we are trying to reduce lots of variation that should not be part of the experiment. These are some of your constants, for example. So in this case, I'm going to take the 4 cores of potatoes, and before I start anything, I want to cut them to the same length. Your lab manual says 2 centimeters. If you go 3 or whatever, it's not going to hurt as long as they fit in the beakers and as long as they are of the same size. Okay, so now I have 4 cores of potatoes that are of the same length. I'm going to drop them in the water for just a couple of minutes to make sure I'm starting with all hydrated potato cores. And when the 2 minutes are over, your lab manual again says 10. That's really too long. We should change that. So a couple of minutes, we're going to take each potato core, we're going to weigh it, and then we're going to drop them in the different solutions. The 2 minutes are over. So now I'm going to take the potato, just roll them quickly on the paper towel to remove the excess moisture. And this is going to go into our water treatment. So the scale is at 0. I place the potato core. And again, please write down this number because we're going to follow this number in 45 minutes. See what happens to that weight. Okay, so good? Yeah, okay. So now this core is gonna go into the beaker that says water in it. Gonna do the same for all of them, and as I said, we're gonna record the weight. So now we're doing the one for the 0.9%. Okay. Okay, this is the one for the 2.5%. And I hope you're realizing, because we cut them to the same size, the weight is almost the same or the same so far. Okay, this is our last piece. My goodness. And this one is gonna go in the 5% sodium chloride. Again, so you have your 4 treatments. We're gonna come back in 45 minutes and weigh them again and see if our hypothesis is true or false for each one of these treatments. Okay, well, 2 hours have passed and now it's time to finish the dialysis experiment. We're gonna take the dialysis tube out. As I said, it's been soaking for a while. We're gonna just remove excess water and we're gonna weigh it and see if there is an increase or decrease in the weight. Okay, so please record this number just as you did before and compare them. So, did your dialysis bag increase in weight, decrease in weight, or did it stay the same? Excellent. It did go up in weight, which means, what does this indicate? This indicates that water went into the dialysis bag, which means this is osmosis occurring. Water went from the beaker, where it's more concentrated, into the dialysis tube, where the water is less concentrated. Again, we're not talking about solutes when we're talking osmosis. We're concentrating on the water molecules. Okay. So this part of the experiment is done. Now we're going to test for dialysis. And the way we're going to do that, if you were in lab and you guys are working in groups, each group would have 4 test tubes, and we're going to use 2 of them to pour water from the beaker. So this is in your lab manual, this is what we call outside. So this is outside water. We're going to see if any of the solutes left the dialysis bag and are now in this water in the beaker. So about the same amount, you don't have to be totally exact. So one of them, we're gonna test for glucose, the presence of glucose, and one, we're gonna test for the presence of starch. Then, we're gonna take the dialysis bag, we're gonna empty the content over 2 test tubes. This is what in your lab manual would say inside. So we're gonna again test for the presence of starch and glucose. Yes, we know there is starch and glucose, but we're still gonna do it to make sure. So the way I do it, just to avoid problems, I always dump everything into 1 test tube, and then I'm gonna split this amount with another tube. And again, we're gonna use 1 to test for starch and 1 to test for glucose. So you're gonna label your tubes properly. So for me, I'm gonna say tube number 1 and tube number 3. I'm gonna use these to test for glucose. So this is for outside glucose and this is for inside glucose. And then we're going to use 2 and 4 to test for starch. The 4 is for the inside starch The 2 is for the outside starch. Again, when I say outside, I'm referring to the beaker, to the water in the beaker. And when I say inside, we're referring to the dialysis tube. Okay, test for starch, we use iodine. And again, iodine has this brownish amber color. And we're going to see if there is starch in the solution, what happens to this color. So I'm going to take my 2 starch, or the tubes I'm testing for starch. This is the one, the outside, meaning the water from the beaker. And we're going to compare them in just a second. This is the inside, and I'm adding again a couple of drops of iodine. And let's see. Immediately, you guys can notice the difference. This is outside. The color did not change. The color of iodine remained the same. It's that brownish amber color. Look in here. Do you see the difference? Inside the tube where we know there is starch, it turned that really dark blue color. So what is this telling us? This is outside. This is the beaker, the water in the beaker. If the color didn't change, what is this telling us? Did starch leave the dialysis tube? No, it did not. Very good. So in this case, starch was not able to cross the dialysis tube. And again, why? Because starch molecules are very large molecules, and the pores in this dialysis tube are very tiny to allow those large molecules to cross. Now for glucose, we're going to use that Benedict solution. And for this reaction to actually take place, if there is glucose that's interacting with Benedict, we need to have our reaction taking place in hot water. So it doesn't occur at room temperature. We need to put these tubes in a water bath. Water bath meaning we can control the temperature of the water, and we usually have it about 80 degrees Celsius. And this is where we're going to see if there is a change in the color. Because it's a little demonstration, I didn't want to turn on the water bath and do all of that. I'm going to create the same environment by putting those tubes in a beaker and putting the beaker in the microwave for a minute. And we're going to see if there is a change in color. But that high temperature is required for us to see any sort of results with Benedict's. So again, I'm going to put a couple of drops. And Benedict, it's one of these solutions that if you were in lab and you're using these tubes, you cannot really dump in the sink. We have to dispose of in a special container because we don't want this to be going into our system. What do you call it? The sewer. The sewer, sorry. Okay, so as you can see, the color is that light bluish color. We're going to put these 2 tubes, and as I said, we're going to go to the microwave, give them, heat the water around them, and we're going to see if there is any change in the color. Okay, guys, we put those tubes in the microwave in water, obviously, for about 1.5 minutes. And now we're going to take them out and see, go to our table and discuss the results. So this is the setup that we started with in the microwave. Okay, so let's see. This is the inside, meaning this is the sample that we took from inside the dialysis bag. Okay, remember, this is the original color, that blue color. We added it to our solution and it turned that deep orange color. Again, it really looks like orange juice. So this change in color indicates the presence of glucose. Yeah, we did put glucose inside the tube, so we expect to see this color. Now, let's see. Remember, we did take a sample from the beaker and we put it in a test tube. And again, this is what we observed. This is the original color. And after, the dialysis tube was soaking for 2 hours and then we added Benedict. So what does this indicate? Does that indicate the presence of glucose? Correct. So that tells us that actually the glucose from the dialysis tube, remember we had the dialysis tube with the solution, that some of the glucose from inside the dialysis tube left the dialysis tube, and now some glucose is found in that beaker, the water in which the dialysis tube was soaking. Again, what is the conclusion? What is the results of this experiment? So if you compare, again, we have the tubes for you, inside and outside. So, sorry, let me put this down. This is outside. We tested for starch and it tested negative. So starch did not leave the dialysis tube. Here we tested inside and outside for glucose, meaning did glucose leave the dialysis tube? It did. So again, if you are asked the question, why did glucose leave the dialysis tube and not starch? Obviously, it's because of their size. Starch is much larger than glucose. Okay, guys, that concludes this particular experiment for you. We're gonna move on and see what happened with our osmosis in potato experiment. Okay, guys, 45 minutes have passed, and now we're gonna start weighing the potato cores in the different beakers, the different treatments that we had. So we're starting with the water. So please write down the weight, and you're gonna compare it to the original weight, and you're gonna come up with your own conclusion and think of the hypothesis that we have discussed previously. So now we're gonna take the next piece of potato, the one soaking in the 0.9% sodium chloride. Now we're gonna weigh it. And again, record the weight and think of the hypothesis and your conclusions. Next, we're gonna move on to the hypertonic ones, starting with the 2.5 sodium chloride. And this is the weight, the new weight. And now we have the last one that was soaking in the 5% sodium chloride. Okay, guys, you have the weight before soaking the potato cores in the salt solutions and after, and also the water treatment. So now your job is to think of your results and again decide whether the hypotheses were true or false, and make sure to answer the questions in the lab manual. And thank you. Guys, this video was prepared to you by Dr. Johnson and Dr. White. Hope you'll find it helpful. Thank you very much.
Thank you.